Welcome to the MCAT Basics Podcast, brought to you by the physicians at Med School Coach. Each week, Sam Smith breaks down high-yield MCAT topics so you can score as high as possible on test day.
Hello, I am Sam Smith, and this is Sam's MCAT Basics. This podcast covers the most important topics put out on the AAMC MCAT each year, and I determined this list by going through all the official practice materials that the AAMC puts out, and also some third-party practice material, and just put together this big list of topics that consistently kept on showing up, and ranked those topics and made podcasts out of it. This podcast covers the reproductive system and development. I'm going to introduce some of the organs in the reproductive system, then talk about spermatogenesis and oogenesis. And then I'll go in and talk about the pathway that sperm take as they grow and mature. And then I'll go into the menstrual cycle, which is kind of, I guess, a little bit the equivalent of the path that sperm take, but only in female. So I guess the path that the egg takes. Next, I'll go into fertilization and development. And here, I'll mostly introduce stuff in a chronological order, and hopefully it will make more sense that way. And this material will show up in one out of the 4 MCAT sections. This will be the bio and biochem section. And it's kind of one of the more important physiology topics or concepts to understand for the MCAT. In addition, it's also a very interesting topic to learn about, at least in my opinion. So I hope this helps, and I hope you enjoy. So there are a few reproductive system organs to know. These are pretty simple, and I'm sure you've heard of these before. The gonads in male are the testes, and in females are the ovaries. Then you have the genitalia, which in men are the testes, the penis, and various ducts and glands that go along with those. Then in females, you have the ovaries, the fallopian tubes, the uterus, and the vagina. And so I'll be using these terms throughout this podcast to describe where events take place, but just know for now that these are kind of the main organs within the reproductive system. All right, so the first thing I want to talk about here is spermatogenesis and oogenesis. So this is the process in which sperm cells, which is spermatogenesis, or egg cells, which is oogenesis, develop. Essentially, we're taking a look at these undifferentiated gametes and looking at the process by which they become the mature sperm and the mature egg. So when meiosis I occurs, you have homologous chromosomes that meet at the metaphase plate and then split apart. And then from there, you have these 2 cells. And in these 2 cells, meiosis II occurs. And what happens there is the sister chromatids line up at the metaphase plate, and then those cells split up. So what happens is you start with 1 diploid gamete, and that becomes 4 haploid gametes. And again, haploid means that these cells have a single set of unpaired chromosomes. So keep in mind the meiosis concept as I talk through spermatogenesis and oogenesis. All right, let's start with spermatogenesis. So the first, most undifferentiated cell we have that will eventually become a mature sperm is called a spermagonia. And you can kind of think of this as it's gonna be a spermagonia. And this is a diploid cell. And so in terms of keeping track of the number of cells we produce, let's just say that we have 1 spermagonia. We're just going to track that 1 diploid spermagonia. So what happens is that spermagonia grows and matures and becomes a primary spermatocyte. And so this primary spermatocyte is also a single cell, and it's also diploid. Next, this primary spermatocyte undergoes meiosis I, and it becomes a secondary spermatocyte. And this creates 2 haploid cells. So you went from 1 diploid cell to 2 haploid cells. Next, these secondary spermatocytes undergo meiosis II, and they become spermatids. And this creates 2 more cells from each of these 2 cells that are each haploid. And so that is a grand total of 4 cells. So from that 1 spermagonia, you create 4 haploid spermatids. these spermatids undergo further differentiation to become spermazoa. And these are the tadpole-like things that you usually think of sperm as. And again, these are haploid cells, and there's 4 of them. So to quickly recap, you start with 1 spermagonia, and you make 4 mature spermazoa. And you might be thinking, okay, more differentiation. What do you mean by that in terms of how this spermatid becomes a spermazoa? Well, essentially, the spermatid is a non-functional spermazoa. It must, number 1, grow a tail and be able to swim, and number 2, get rid of excess cytoplasm in order to become functional. And both these changes have to do with making the sperm mobile. Let's quickly compare this to a swimmer. You wouldn't want a really good swimmer to have no arms, right? So that's why you need this spermazoa to have a tail, right, in order to be able to swim. Kind of like, it's like a swimmer swimming with no arms, right? That's not going to work. And then what happens when this spermatid gets rid of the extra cytoplasm, it becomes kind of like more aerodynamic in the water, right? It goes from this round shape to this skinny, more like fluidly dynamic shape. And so if you think of an Olympic swimmer, you don't think of like a very fat, round person that becomes a swimmer, right? Because that's extra surface area means extra drag as you move through a fluid, and therefore, you got to put in more energy. You don't go as fast, and that is why you need to get rid of that extra cytoplasm within the spermatid. So that is the process by which a spermagonia becomes a primary spermatocyte, then a secondary spermatocyte, then a spermatid, and then all the way to a spermazoa. And another thing that's important to understand is where these events take place. And so let's go over that real fast. So spermatogenesis takes place within the seminiferous tubules, and these are inside the testes. It's important to note here that there are 2 types of cells that help the sperm grow. The first are called serotoli. The next are called Bertolli. And so the serotoli cells nourish growing sperm in the seminiferous tubules, help them grow. And then Bertolli cells actually produce Alfredo sauce.
Bertolli.
Just kidding. The other type of cells that are important to know that reside within the seminiferous tubules are called cells of Leydig, and they actually produce testosterone. So, the serotoli cells help nourish growing sperm in the seminiferous tubules. That is step 1 in the maturation pathway. And then the sperm pass into the epididymis once they gain motility and become spermazoa, and they are stored in the epididymis till ejaculation. And then during ejaculation, the sperm travel down the vas deferens and into the ejaculatory duct. From there, the 2 ejaculatory ducts fuse to form the urethra, with which the sperm is ejaculated through. And there's an easy way to remember all these different locations within the body the sperm travel, as well as the chronological order in which they travel through these different parts. And that acronym is 7UP. So you have seminiferous tubules, epididymis, then vas deferens, then ejaculatory duct. N is nothing. U is urethra. And then P is penis because it travels out the penis. So don't forget 7UP. And before I get into menstruation and talk about female oogenesis, I briefly want to mention the role of luteinizing hormone and follicle stimulating hormone in men, right? Because a lot of times we think of this strictly in terms of menstruation. A lot of times we don't think that these do anything in men, but they do. So in men, follicle stimulating hormone stimulates serotoli cells to produce spermagonia. And obviously these play a big role in becoming sperm. And then luteinizing hormone, on the other hand, stimulates cells of Leydig to produce testosterone. So it's important to remember FSH corresponds to serotoli cells, LH corresponds to cells of Leydig, in men, of course. All right, so now let's switch over to the female side and talk about menstruation, and then get into oogenesis. So what is the menstrual cycle? So the menstrual cycle is broken down into 4 phases. Number 1 is the follicular phase. Number 2 is ovulation. Number 3 is the luteal phase. And number 4 is menstruation. A lot of important events occur during the follicular phase. Number 1, follicle-stimulating hormone is secreted by the anterior pituitary gland. And this follicle-stimulating hormone, or FSH, induces the proliferation of granulosa cells. And what happens next is these granulosa cells begin to actually secrete estrogen. And this increased level of estrogen that is secreted by granulosa cells stimulates the production of gonadotropin-releasing hormone, which this gonadotropin-releasing hormone goes on to increase the production of luteinizing hormone. So you will see essentially a little bump up in levels of gonadotropin-releasing hormone, and that stimulates the LH spike, or the luteinizing hormone spike. And so this LH surge plays a huge role in ovulation. So to quickly recap everything, there's 2 big events in the follicular phase. Number 1, you have this huge increase of LH that becomes a peak right before ovulation. You have a slight increase in the amount of gonadotropin-releasing hormone, and you also have a big increase in the amount of estrogen. So that's kind of the 3 things to remember. All right, now into ovulation. As I said, ovulation is triggered by a luteinizing hormone surge. And ovulation is when a mature egg is released from the ovary and pushed down into the fallopian tube and made available to be fertilized. And the lifespan of this egg is typically around 24 hours. Unless it meets its good friend Sperm Jones during this time, then it will die. So that's the ovulation phase. Now we get into the luteal phase. And so during ovulation, the egg bursts from its follicle, and this ruptured follicle stays on the surface of the ovary. And for the next 2 weeks or so, the follicle transforms into a structure known as the corpus luteum. And this structure starts releasing progesterone along with very small amounts of estrogen. And this combination of hormones maintains and thickens the lining of the uterus while it waits for a fertilized egg to stick. And I think you can remember which hormone does what by thinking progesterone protects the endometrium and estrogen establishes the endometrium. So you need estrogen in order to establish that corpus luteum. And then once the corpus luteum is established, it secretes progesterone, which maintains the endometrium, which helps it grow. And so if pregnancy does not occur, the corpus luteum withers and dies away usually around day 22 in a 28-day menstrual cycle. It's important here to understand that the corpus luteum is what is left of the follicle after the secondary oocyte is released during ovulation. And it remains in the ovaries as the egg travels to the fallopian tubes. So essentially, this corpus luteum acts remotely, right? It's kind of hanging out in the ovaries still and releases these different hormones that affect the endometrium. The last phase is the menstrual phase, and this occurs if a female is not pregnant. If a female is pregnant and has a fertilized egg, this part will not happen. And so what happens during the menstrual phase is progesterone levels fall off, and this causes the lining of the uterus to slough off, and this is also known as a period. So there are a few times to know in terms of the menstrual cycle. So for instance, at 14 days, the LH or luteinizing hormone spike occurs, and again, this is when the egg is released. And then at day 28, one cycle ends and the next cycle begins. And this is why we think of this as a monthly cycle. And so you always hear about those people that are against contraceptives that talk about timing sexual intercourse with the cycles of the moon or whatever it is, or the cycles of the waves of the ocean, whatever it is. There's times that you can have sex and not worry about getting pregnant. That is obviously during the luteal phase when an egg has not yet been implanted, and then also during menstruation. So that's something to keep in mind. There is a period of time in which pregnancy cannot happen due to an egg not being in the fallopian tube, being there ready for fertilization. All right, so that is the menstrual cycle. I want to get into oogenesis now. So I want to answer the question, how does the egg develop? We talked about spermatogenesis. Now I'm going to talk about oogenesis. And there's some similarities, there's some differences. Naming is very similar. So we start with the oogonia, which is going to become a mature egg. And that is one diploid cell. That oogonia grows and matures and becomes a primary oocyte. And then this primary oocyte undergoes meiosis 1 and is split into two cells. And this is where we start to see differences between spermatogenesis and oogenesis. So one of these cells that is produced from the primary oocyte is called a polar body. And you can think of this like the runt of the litter. This is a small cell, is non-functional, really teeny, like no cytoplasm, and just kind of dies off. The other cell becomes a secondary oocyte. And at this point, that cell is haploid. So this remaining secondary oocyte undergoes meiosis 2 to become another polar body, which dies off, and an ootid, which is also haploid. So this ootid matures and differentiates then to become an ovum. And so you can see here, we're going from one oogonia to one ovum. And so in this case, you have two polar bodies that kind of die out at the end of meiosis 1 and meiosis 2. So that's kind of the main difference between spermatogenesis and oogenesis. So to me, though, what's really interesting about oogenesis is when these processes all take place, right? It's not just this linear straight line from a oogonia to primary, secondary, ootid. It's not just this linear thing that happens, 5 minutes between each cell. It's not how it works. This is what makes this process so interesting, in my opinion, in terms of producing an ovum. So by birth, all oogonia have undergone DNA replication and are primary oocytes. And these primary oocytes are arrested at prophase 1. And remember, these are diploid cells. And so I'm sure you've heard that all women have all their eggs by birth. And this is true, right? You have all these oogonia at birth that have undergone DNA replication and are primary oocytes. And then once a month, a few primary oocytes will finish meiosis 1 and become secondary oocytes. And at this point, the secondary oocytes are arrested in metaphase 2 during meiosis 2. And these secondary oocytes, again, are haploid. And then, if one of these secondary oocytes is lucky enough to be fertilized by our friend Joe's sperm, then what happens is this secondary oocyte completes meiosis 2 and produces the haploid ootid. And then, of course, the ootid matures to the ovum, and this happens in a few minutes. And at this point, this ovum is still haploid. And so quickly, to compare the differences between spermatogenesis and oogenesis, there are three that I'm going to mention. The first is that oogenesis produces these non-functional cells known as polar bodies that just kind of disintegrate and go away. And therefore, one oogonia eventually produces only one egg. In opposition to that, one spermagonia produces 4 mature sperm cells. Number 2, men constantly produce sperm. This is in contrast to women who have a set amount of eggs when they are born. And then lastly, oogenesis is partly dependent on sperm fertilization, whereas sperm development does not have any dependency on an egg. So let's quickly mention an interesting case in which the application of women having all their eggs at birth plays a role. And that is in women having a greater risk of having children with trisomy 21, which is Down syndrome, as they get older. And so Down syndrome is caused by a random error in cell division that results in the presence of an extra copy of chromosome 21. This type of error is called a non-disjunction, and this usually occurs when one cell divides in two and pairs of chromosomes are split so that one pair of chromosomes goes to one cell and the other pair goes into another cell. However, in non-disjunctions, chromosomes from one pair go into one cell and no chromosomes go into the other cell. So essentially, you get two pairs of a single chromosome into one of these cells. And kind of interestingly, virtually all of the mitotic errors of chromosome 21 that result in Down syndrome seem to be initiated in mitosis 1. And most of these errors occur in the eggs, not the sperm. Now, it's at this point that I want you to remember that meiosis in a woman extends over a 10 to 50 year period, right? With the oocyte being arrested in meiosis 1 during most of its lifetime. So more time between fertilization equals more time for bad things to happen to that egg, right? It's just sitting there arrested in metaphase 1. So the longer it just kind of sits there, there's a higher probability that these random errors occur that could result in trisomy 21. And there's been a few different mechanisms that have been proposed for why non-disjunctions seem to occur after a longer period of time. And this is from a 2006 review, so the literature might be more updated by now, but this is a little bit older review. So one of the mechanisms proposes that an accumulation of toxins from the environment during the arrested state of the oocyte leads to this non-disjunction. A second mechanism that's been proposed is that a degradation of mitotic machinery over time while in the arrested state leads to a suboptimal resumption of meiosis 1 and meiosis 2. And the third one says that a change in ovarian function due to suboptimal hormone signaling might lead to that non-disjunction. And likely it's a combination of some of these. Maybe each case is different, but just kind of interesting, something to think about. So that concludes the introduction into spermatogenesis and oogenesis. All right, so I want to get a little more into development now. And let me just quickly recap where we're at. So we are at the point in which a female has had a luteinizing hormone surge, which causes ovulation, and that egg is released down into the fallopian tube. And then this female has had sexual intercourse with a male, and the sperm has made its way up to the fallopian tube where it fuses with the egg. And what this does is this causes the egg, which is at this point a secondary oocyte, to now become an ootid, which then develops into an ovum. Now, a few quick interesting asides. Number one, it takes a bit of time for the sperm to actually become mobile when it's in the vagina. This is something I didn't know, is that a sperm must undergo a process called capacitation in the female's reproductive tract over a period of a few hours, which eventually increases its motility and destabilizes its membrane to prepare it for what is called the acrosome reaction. And I'll get into that in a sec. Another interesting thing here is that spermicide, which is an active ingredient in condoms, for instance, what it actually does is it inhibits sperm motility. And so say sperm was a swimmer, well, the spermicide essentially acts to cut off the arms and the legs of the swimmer, rendering it immobile. All right, let's talk about what happens when sperm reach the egg. And this stuff's pretty interesting. So at the beginning of the process, as I talked about, the sperm that enter the vagina must undergo capacitation, making them essentially mobile. This takes a few hours. Once this happens, the swim make their way up to the fallopian tubes, where the sperm bind to what's called the corona radiata, which is a layer of follicle cells on the outside of the secondary oocyte. And so essentially what this looks like is a bunch of smaller little cells piggybacking off the bigger secondary oocyte. And if you don't really know what I'm talking about, just Google a picture. It's kind of interesting. Google corona radiata, just see what these cells look like that kind of surround the egg cell. And at this point, a bunch of sperm bind, right? You got the sperm coming in from the left, whizzing by on the right, ting, 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 ting. All these sperm are hitting the egg and binding to that corona radiata. After this binding to the corona radiata occurs, the sperm reach what's called the zona pellucida, which is an extracellular matrix made of glycoproteins. What happens when the sperm reach the zona pellucida is that this triggers the acrosome to burst, releasing enzymes that help the sperm get through this zona pellucida layer. And you might be thinking, okay, what's an acrosome? Well, an acrosome is an organelle that's on the top of the sperm, and it kind of looks like a helmet. So just go back to the swimmer example. Imagine a swimmer wearing a helmet, and this helmet helps them break through walls. Essentially, that's kind of how this acrosome works. And these acrosomes contain digestive enzymes that essentially break through this glycoprotein layer that the zona pellucida makes up. And then once these sperm find their way past this zona pellucida, the cortical reaction occurs. And this is when cortical granules in the secondary oocyte are released and make their way to the exterior of the cell. And then these granules fuse with the cellular membrane and they release their contents. This is exocytosis. And then this causes the glycoproteins in the zona pellucida to actually cross-link and become this immobile, impenetrable brick wall, if you will, so that other sperm can't get in. And, why does this happen? Well, this happens because you want to make sure only one sperm gets in and fertilizes that egg. So essentially, you can imagine the sperm swimming in and it squeezes through and slides right in. And right as this happens, the zona pellucida cross-links and becomes this brick wall that no other sperm can get through. So now the sperm has made it in. Hopefully it's the only one. In the case of twins, as I am, sometimes you get 2 sperms into 1 egg. But let's just say in this case, only 1 sperm made its way in. Well, now what happens is that after the sperm enters the cytoplasm of the secondary oocyte, the tail and the outer coating of the sperm actually disintegrate. And then the oocyte now undergoes its second meiotic division, producing the ootid and then also releasing a polar body. And at this point, the sperm nucleus then fuses with the ovum, and together you have a diploid cell, and then this starts to develop into a little baby. And kind of a little interesting note is during the step in which the tail and the outer coating of the sperm disintegrate, there is another part of the sperm that actually also disintegrates. And think about it. What other part of a cell, or what other part of inheritance come directly from your mother? What would make sense to be degraded just in the male, but not in the female cell? And the answer there is the mitochondria. So at this same stage, the male mitochondria actually disintegrate, and this is why you inherit only your mother's mitochondria. So once this has all gone through, you've had fusion of the 2 genomes. The cell starts to divide mitotically, undergoing mitosis, and is now called a zygote. So that's a pretty cool process, what happens in the sperm as they make their way up to the egg and break through and everything like that. Pretty interesting. So once you have this zygote formed, you start having cellular differentiation. These cells start specializing, start forming different tissues, etc. And I'm actually going to do a bonus episode on cell differentiation. It's super interesting, and I didn't really have enough time to really talk about it in full length as I really wanted to. So look out for a bonus episode on cell differentiation. What I'm going to talk about next is a few different processes of development that you should know. So what you really need to understand here are the different stages of embryonic development. Kind of know the names, know the timelines of when they happen. This is something I've definitely seen on practice tests and stuff. So, as I as I left off from earlier, right, we have this zygote. And so the zygote is a solid ball of cells, and this solid ball is called a morula. And what happens is this morula undergoes blastulation to form a hollow ball of cells, and that morula is now called a blastocyst after it's formed this hollow ball. And that typically happens about 5 to 8 days after fertilization. This blastocyst then implants on the wall of the uterus. Typically, this happens about 9 to 10 days after fertilization. After this, gastrulation occurs. And what happens here is you have an invagination of the blastula. And you can imagine this as kind of a folding over of the wall of this blastocyst to form an invagination. And eventually that invagination becomes the anus. Another important thing that happens during gastrulation is that the germ cell layers form, and that is the mesoderm, ectoderm, and endoderm. And I'll discuss those next. Definitely another kind of topic that you'll see is they might say something like, what germ cell layer is the kidney from? Or, or what is germ cell layer is the hair from? Just stuff like that. So anyways, just know that this happens during gastrulation, and gastrulation occurs around the 3rd week after fertilization. The next kind of stage is called neurulation. And in neurulation, you have a folding process whereby the neural plate is transformed into the neural tube. And this takes place during the 4th week. And again, this is sort of like an invagination, except you actually form this tube that becomes the neural tube. This takes place at or during the 4th week after fertilization. So those are the 4 kind of main stages of embryonic development, or main early stages of embryonic development to know. The next thing that's important to know are the different germ layers. So what I'm going to do is I'm going to talk about the 3 germ layers and then talk about the different organs or different parts of the body that are developed from each of those layers. So first of all, let's define a germ layer for you. So a germ layer is a primary layer of cells that forms during embryonic development, again in the gastrulation phase. And there are 3 types of germ layers that you should know, right? And I mentioned these briefly. Is the mesoderm, the ectoderm, and the endoderm. And so let's start with the mesoderm. So the mesoderm consists of essentially the muscles and circulatory system, and specifically mainly consists of the circulatory system, muscles, and the excretory system. And you can kind of think of this as mesoderm, muscle. That's kind of how I think of it. Anyways, the mesoderm contains the blood vessels, lymphatic vessels, spleen, gonads, kidneys, muscles, connective tissue, circulatory system, and excretory system. The ectoderm, on the other hand, contains things that make you attractive. So what makes people attractive? And this is kind of how I remember it. So first of all, your brain makes you attractive, and let's hope that that's the case for most people, at least if you're not shallow. So neurons are developed from the ectoderm germ layer. Skin is developed from the ectoderm germ layer, right? Because skin's pretty, people find that attractive. And then lastly, hair, teeth, and eyes are all things that people find attractive and are also derived from the ectoderm. So again, that's neurons, skin, hair, eyes, teeth. And last but not least is the endoderm. And when I hear endoderm or see endoderm, I like to think of inner layer or lining. So you hear endo, think inside. And that's because the digestive tract and the lungs are both derived from this endoderm germ layer. And in addition to the digestive tract and lungs, you have the parathyroid, the thyroid, the pancreas, the liver, and the thymus. And so, put these on note cards, whatever you have to do to memorize them, write them down a bunch, whatever, but they are important to know. And, there are some tricks, obviously, that I just mentioned to kind of help you memorize them, but those don't apply for every single tissue or organ that is derived from that specific germ layer. So make note cards, figure it out. But good stuff to know. All right, so where are we at with everything now? We essentially left off at the stage in which neurulation has occurred and this baby is starting to develop. And what I want to do is I want to skip forward a bit to when that baby is fully developed. So let's skip forward. So I just fast forwarded, and now we're at the point in which the baby is fully developed and ready to come out. And so what happens? Well, first of all, the body's got to know that it's time to give birth. So how does that happen? Well, from what I could read and what I gather is that it's thought to be because of changes in levels of different hormones, and that's specifically estrogen. So you have a peak of estrogen in the body right around week 40 of pregnancy, which pregnancy lasts 40 weeks for guys out there who don't know. And what happens then is you get an increase in estrogen, you get a drop in progesterone. And what's thought to happen then is that this process actually activates these contractile cells that are sensitive to oxytocin. And so now these cells become sensitive to oxytocin, which is the hormone that actually makes the uterus contract and that baby come out. And so this is kind of the process by which people believe that the body knows it's time for that baby to come out. And what happens around this time too is that the baby settles its head down lower in the uterus. And this causes the cervix to start to dilate. And then at this point, the uterus starts to contract. Again, this is stimulated by oxytocin. These cells have been activated by an increase in estrogen, is the thought. And it's important to note that oxytocin action is a positive feedback loop, right? As you release more oxytocin, you get more contractions, and more contractions cause more oxytocin to be released. And you just get this kind of snowballing effect in terms of contraction and oxytocin. And then you have birth, and the baby comes out. And from here, I really can't offer any more advice. I believe that you got to keep chocolate away from babies, and you got to make sure that you have the right kennels. Other than that, I'm not really sure what the next steps are. But what's important to understand here are the process of birth and development of the fetus. And so this is kind of a basic introduction to the reproductive system, development, and then even a little bit on birth. I hope this helps. I try to keep it kind of like a story almost, and I hope that kept your attention. And listen to this a few times if you need to. But this information is important for the MCAT. Thanks as always for listening. If you found this helpful, go give us a review on iTunes. If you didn't like it, shoot me an email. Just say, hey man, this isn't right, or this is right, or even if you do like it, I always like to see emails of people saying, hey, this really helped. I'm taking my test this week. It's always good to hear feedback. So, thanks for listening. I'll see you for the next one. All right, now for a new segment that I'm going to call Indecent Idea of the Day. And this is related to reproduction. It is also R-rated, so please shut this off if you do not have your parents' consent to listen to this. So let's talk real briefly about female copulatory vocalizations, also called female copulation calls. And these are produced by female primates, including human females, and female non-primates. And if you don't know what I'm talking about, I'll put this in layman's terms. I'm talking about sex noises. And so if you get to kind of thinking about sex noises, you might be wondering, do these play some kind of evolutionary role? Why are they conserved through primates, non-primates, and also humans? Like, do they play some weird role? And the answer to that is yes. I came across this article, scientific article, talking about some of the theories behind some of the theories that they believe in terms of the evolutionary role that sex noises play in terms of giving some sort of advantage to the female, or some kind of advantage to the female's offspring. So there's 3 different theories I'm going to present. The first is that these vocalizations can be used to intentionally make these females' partners ejaculate more quickly when they use these vocalizations. So what would be the point there? Well, the point there would be, let's say that this female monkey is mating with a male monkey, and this female really wants to get pregnant by this male. This is, the head of the tribe. She'll do anything she can in order to get impregnated by this male monkey. So she uses this vocalization, right? If this vocalization would make this other male ejaculate quicker, then there'd be a higher probability that this female monkey would become pregnant. Kind of interesting. Theory 2 says that female monkeys use these sex noises in order to attract other males, resulting in multiple males having sexual intercourse with this monkey. And then when the child is born, none of these male monkeys know whose child it actually is. And so this actually increases the chances of that child monkey of living longer, right? It reduces the risk of attacks against that small monkey, right? If all these males think that, okay, maybe I'm the dad. No, you're the dad. No, I'm the dad. No, I'm the dad. Then there's less chance of this little baby monkey being attacked. And also these adult monkeys are more apt to act reciprocally towards this baby. They're more apt to give food, to give water, etc. So that's just two kind of theories on the evolutionary value behind female copulatory vocalizations. Kind of interesting and related to development, so I thought I'd bring it up.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.
Come on, Murph. I'm trying to podcast over here, dude.